Hi class, I'm Mrs. Climo, and we're here to
I'm Mrs. Jack, sorry.
give you a little preview of our lab for today.
Today's essential question is, how can DNA be extracted? And in order to demonstrate that, we're actually going to do a strawberry DNA extraction.
All right. Well, to begin with, as you know from the other day when you went to the lab and were able to access that video, you can actually take DNA from any substance. But strawberries work especially well.
Right, and in order to do that, other than strawberries, you definitely need an extraction buffer, and we're actually going to show you what we did to make the extraction buffer. We used salt, dish detergent, and distilled water. We added all those together, a specific amount, and we came up with the extraction buffer that all of you will need today in order to extract DNA from your strawberry.
Additionally, we have centrifuge tubes, 2 different sizes, a large one and a small one.
We have cheesecloth that is going to act as a second filter, or actually I say a filter to our funnel. We'll be placing that inside the funnel, and we'll talk about that in a little bit.
Additionally, at the very end of the process, you'll be using this isopropyl alcohol, which must be chilled, or ethanol. So it's chilled. Also, we have our bamboo skewer that you can use at the end of this activity to actually wrap the DNA around it.
I think we're ready to get started.
Okay, so if you look at our focus for today, it asks, what is DNA? Hopefully everyone knows.
And of course, what does it look like?
That is the next part. So here's our procedure. Within your lab group, we're going to get you a strawberry in a baggie, and you want to gently mash it.
Oh, no, just
Not
kidding.
like that.
So we're going to mash like this, just nice and gently. Did we mention we want to make sure that this was free of air? So make sure you squeeze all the air out first.
Okay, mash away. The directions say about 2 minutes. We want it really smashed. Okay? Good job, Mrs. Jack.
Thank you very
Doing a
much.
fine job. While Mrs. Jack is smashing, let me just ask you this. Why would our extraction buffer contain just soap? Why would we need that? How is that going to help us break down our cell membranes? Because really, that's what that buffer is going to do. It's going to break down those cell membranes to allow that DNA to get out of the nuclear membrane and out of the cell membrane. What's in that cell membrane again that doesn't like water?
Carbohydrate?
No.
Protein?
Lipid. Lipids. All right, so lipids can be broken down with some dish soap. And those lipids, remember, are part of the cell membrane.
Right, think about when you have a really greasy dish, what do? You use soap in order to break down the oil. So same thing happens there.
And here's a cool thing. This is called lysing the DNA. Wonder if it has anything to do with the lysosome.
Pretty cool.
All right, so that looks pretty mashed up. At this point now, we're going to measure out our extraction buffer. Okay, so if you want to, I think I'm going to pipette mine in. All right, so you're going to need, how much, Mrs. Jack?
10 milliliters.
10 milliliters, and these are graduated. All right, so go ahead and put your 10 milliliters in.
And once we have our 10 milliliters, we're going to go ahead and open our bags up, and we're going to pour this right in. So the extraction buffer will be with the strawberries, and we'll gently mix these together. You want to be careful when you're doing this. You don't want to be crazy and agitate it. You don't want a bunch of air bubbles, so just be very gentle with it.
Yeah, I'm making too many bubbles.
That's okay.
Okay, so I have my 10 milliliters of extraction buffer.
We're going to go ahead and make sure we reseal. And again, just kind of pressing it lightly. You don't want to get too many bubbles. Some are okay, but if you get too much, it's really going to impede your results. Okay.
So after a minute of smushing,
We're needing to set up our filtration. And the filtration is very simple. We're putting our cheesecloth in the funnel, and that's going to help us do what? Think about what we will be doing with that. I'll give you a second. Did you get it? Hopefully you said that it's going to strain the strawberry, and that's exactly what we're doing next. We're pouring this solution in very carefully. Again, we don't want to agitate it. We don't want a lot of bubbles. Okay? Now this process will take 3 to 5 minutes, but apparently the magic of TV, it's happened instantly.
we had a pretty juicy berry, so we have You really you need just the bottom of this to be filled, because you only need a few milliliters. 2 milliliters actually, so that is really more than enough. Once you have enough, you can go ahead and set your funnel aside in your little dish here. I think I'm going to have cut cheese pockets, but okay. So now we only need 2 milliliters. So we want to carefully pour. Remember, these are graduated. Carefully pour your 2 milliliters in. Good job, Mrs. Jack.
Almost there. Okay.
All right, now this is where the alcohol is going to be used. All right, the alcohol has to be kept cold, so Mrs. Jack and I have that on ice up here, and we will be around with the alcohol, and you can pipette out what you need. Then very slowly, you want to let it slide down the side of your centrifuge tube. My fingers are in the way. All right, because the alcohol is going to float on top of the strawberry, and then very, very quickly, you're going to be able to see some results. You'll be able to see the DNA coming out of solution. What else, Mrs.
That's
Jack?
basically it. So after you do this, you'll be able to answer the conclusion questions, and they're very difficult, so you have to really think about them. You'll be thinking about why we used the detergent, why we used the alcohol, why we basically can use DNA extraction as a way to help us in our outside world. So you'll be thinking about all that. And I guess that's a wrap.
Oh, at this point, you can use your bamboo skewer after your DNA starts to form, and if you want to wrap it and spool it around your bamboo skewer, feel free.
That's it.
That's it.
All right.
See how bad this is?
or wait, even better yet, it didn't record. No, it did.